What's up, YouTube? I'm back with another video for you all. This is my last video of my human physiology. I got out of my class with a B, so overall, I feel like my human physiology notes and everything else went a lot smoother this time. Helped me a lot more on the test, so I would suggest you all keep up on these if you're taking physiology, and if you do anatomy, and keep up on the parts of everything. That'll help you a lot. Maybe not as much as my lecture exams, or my lecture exams won't help as much as the parts of the body. So here we go. I'm going to dive right into it. Comment, like, subscribe. Reproductive system. Male reproductive system. Organs are designed to produce sperm and hormones. Transport sperm to eggs. Primary sexual organs, gonads, are testicles. Produce sperm and hormones. Secondary sexual organs have a supportive function. Male reproductive system are organized to produce sperm, produce hormones, and transport sperm to eggs. Primary sexual organs, the gonads, are the testicles, which produce sperm and hormones. Secondary sexual organs have a supportive function. Testes, seminiferous tubules where spermatogenesis occurs. What are the cells that do this? Sertoli cells. Seminiferous tubules contain receptors for follicle-stimulating hormones. Testes, seminiferous tubules where spermatogenesis occurs. Sertoli cells are the cells that help spermatogenesis. Seminiferous tubules contain FSH, follicle-stimulating hormones. Interstitial Leydig secrete testosterone, contain proteins for luteinizing hormones. Interstitial Leydig cells secrete testosterone and contain receptor proteins for luteinizing hormones. Testes, what makes up the testes are the seminiferous tubules in Leydig cells. Leydig secretes testosterone. Seminiferous tubules is where spermatogenesis occurs. Testes, seminiferous tubules in Leydig cells. Testosterone secretion, an initiation between initiation and maintenance of body changes during puberty. Growth of muscles, larynx, and bones. Hemoglobin synthesis acts in paracrine fashion to support spermatogenesis. Testosterone secretion, initiate and maintenance of body change during puberty. Growth of muscles, larynx, and bones. Hemoglobin synthesis acts in peregrine fashion to support spermatogenesis. Sertoli and Leydig cells secrete small amounts of estradiol. Sertoli and Leydig cells secrete small amounts of estradiol. Can males secrete estrogen too? Yes, because they have Sertoli and Leydig cells that secrete small amounts of estradiol. Estradiol is the most form of estrogen in male and females. Estradiol is the most common form of estrogen in males and females. Male accessory organs, epididymis, prostate, and seminal vesicles, bulbourethral gland. Male accessory organs are epididymis, prostate, seminal vesicles, and bulbourethral gland. Epididymis moves sperm from testes into epididymis where they mature and store. Epididymis moves sperm from testes to epididymis where they mature and are stored. Prostate secretes seminal fluid, the fluid that nourishes and transports sperm. Prostate alkaline fluid is important to neutralize acidity. The prostate secretes seminal fluid, alkaline fluid, which is important to neutralize acidity. Seminal fluid that the prostate secretes is the fluid that nourishes and transports the sperm. Seminal vesicles, fluid that will form part of semen. Seminal vesicles secrete fructose. Seminal vesicles secrete fructose. Seminal vesicles is fluid that will form the part of semen, or form part of semen. What is the bulbourethral gland? What does the bulbourethral gland do? It neutralizes acidity of urethra and vagina, provides lubrication for tip of penis during intercourse. Bulbourethral gland neutralizes acidity of urethra and vagina, provides lubrication for tip of penis during intercourse. Erection and emission and ejaculation. Erection, increased vasodilation of penile arterioles. Blood flows into erectile tissue of penis. Erection, increased vasodilation of penile arterioles. Blood flows into erectile tissue of penis. Emission, movement of semen into urethra. Emission is movement of semen into urethra. Ejaculation is forcible expulsion of semen from urethra out of the penis. Ejaculation is forcible expulsion of semen from urethra out of the penis. Normal volume of ejaculate is 1 to 5 milliliters. Normal volume of ejaculate is 1 to 5 milliliters. 60 to 150 million sperm per milliliter. 60 to 150 million sperm per milliliter. That's important to know. Female reproductive system. The organs of the female reproductive system are specialized to produce and maintain the eggs, transport these cells to the site of fertilization, provide a favorable environment for the developing fetus, give birth, produce female sex hormones. Female reproductive system. The organs of the female reproductive system are specialized to produce and maintain the eggs, transport these cells to the site of fertilization, provide a favorable environment for the developing fetus, give birth, produce female sex hormones. The primary sexual organs are the ovaries of the female reproductive system. They produce eggs and secrete hormones. Primary sexual organs of the female reproductive system are the ovaries. They produce sperm and secrete hormones. The other parts of the system comprise external and internal accessory secondary sexual organs of the female reproductive system. The secondary sexual organs of the female reproductive system are the fallopian tube, uterus, vaginal canal, labia, glands for sperm, and breast external. The secondary organs of the female reproductive system, sexual organs of the female reproductive system, are fallopian tube, uterus, vaginal canal, labia, glands that cleanse for sperm, and breast. Describe the process of ova-egg development. How many immature eggs are we born with? How many mature? Primary follicle, Graafian follicle, into oocyte, ovulation, then into ovulation. Corpus albicans is what is called when the egg is recycled. Women are born with 2 million oocytes. Their 400 oocytes are ovulated during the reproductive years when they mature. Describe the process of egg development. How many mature eggs are women born with? How many mature? Primary follicle, Graafian follicle, oocyte, ovulation. Corpus albicans is what happens. Oocyte is what happens right before ovulation. Ovulation is when the egg becomes ready to move on. Corpus albicans is what happens when the egg doesn't develop. Born with 2 million oocytes, 400 oocytes are ovulated during reproductive years as women mature. What is the significance of the follicle fertilization of the egg? The follicle breaks open and releases the egg from ovary for fertilization. What is the significance for follicle during fertilization? Follicle breaks open and releases egg from ovary for fertilization. Where does fertilization occur? In the fallopian tubes. Where does fertilization occur? In the fallopian tubes. What is the menstrual cycle? What happens at day 14? What responds to FSH? What does LH do? The menstrual cycle is a series of natural changes in hormones, production, and structures of the uterus and ovaries of the female reproductive system that makes pregnancy possible. Ovarian cycle at day 14, ovulation occurs. FSH, follicle stimulating, will mature the follicle, help grow the egg. LH helps control the menstrual cycle and triggers ovulation. The menstrual cycle is a series of natural changes in hormones, production of structures of uterus and ovaries of the female reproductive system that makes pregnancy possible. Ovarian cycle at day 14. Ovarian cycle. At day 14, ovulation occurs. FSH, follicle stimulating hormone, will mature the follicle and grow the egg. LH helps control the menstrual cycle, triggers ovulation. Menopause is cessation of ovarian activity and menstruation at 50 years. That's standard. Ovaries are depleted of follicles, produce no estrogen. Luteinizing hormone and follicle stimulating hormone are high because of no negative feedback. Lack of estrogen from ovaries is most responsible for hot flashes and osteoporosis, and increased risk of atherosclerosis. Menopause, cessation of ovarian activity and menstruation at 50 years. That's about standard, 45 to 55 years. Ovaries are depleted of follicles, no estrogen. Luteinizing hormone and follicle stimulating hormones are high because of no negative feedback. Lack of estrogen from ovaries is most responsible for hot flashes, osteoporosis, and increased risk of atherosclerosis. Pregnancy is the presence of developing offspring in the uterus. An event resulting from fertilization. It involves transport of sex cells, fertilization, embryonic development, hormonal changes during pregnancy, and other changes during birth. Pregnancy is the presence of developing offspring in the uterus. An event resulting from fertilization. It involves transport of sex cells, fertilization, embryonic development, hormonal changes during pregnancy, other changes during pregnancy and birth. What do you know about these stages? During pregnancy, you have positive feedback for hormones. During pregnancy, you have positive feedback. Your hormones are increased. Mammary glands. The mammary glands are accessory organs of the female reproductive system that are specialized to produce and secrete milk after pregnancy. The stimulus for milk secretion is oxytocin, myoepithelial cells. Oxytocin is the stimulus for milk production. The nutrients in breast milk are better absorbed and used by your baby. Mammary glands. The mammary glands are accessory organs of the female reproductive system that are specialized to produce and secrete milk after pregnancy. What is the stimulus for milk production and secretion? Oxytocin. Breast milk. The nutrients in breast milk are better absorbed and used by your baby. Immune system. Review 5 types of white blood cells and their basic functions. Neutrophil, eosinophil, basophil, monocyte, lymphocyte are 5 types of white blood cells. Neutrophil, eosinophil, basophil, monocyte, and lymphocyte are 5 types of white blood cells. Neutrophils are phagocytic. Eosinophils detox. Eosinophil detox. Basophil releases an anticoagulant called heparin. Basophil releases an anticoagulant called heparin. Monocyte phagocytic. Lymphocyte provides specific immune response called antibodies. Lymphocyte provides a specific immune response called antibodies. Neutrophil, eosinophil, and basophil are granular. Monocyte and lymphocytes are agranular. Defenses, non-specific or innate, special or acquired. Defenses, non-specific or innate, special or acquired. Non-specific or innate lists the elements in external and internal. Non-specific or innate lists the elements external and internal. Specific or acquired due to prior exposure lymphocytes, mediated by antigens and antibodies. All the structures and processes that provide a defense against pathogens. Specific or acquired due to prior exposure lymphocytes, mediated by antigens and antibodies. All of the structures and processes that provide a defense against pathogens. Non-specific immunity includes the inherited barriers to penetration and body of the body and internal defenses that follow. External defenses: skin, GI tract, respiratory tract, urinary tract, reproductive tract are all external defenses. The epithelial lining of these areas is the main line of defense. Non-specific immunity include the inherited barriers to penetration of the body and internal defenses that follow. External defenses: skin, GI tract, respiratory tract, urinary tract, reproductive tract. The epithelial lining of these areas is the main line of defense. Internal non-specific defense: phagocytosis, fever, or endotoxins, and interferons. Internal non-specific defenses are phagocytosis, fever, and interneurons. Fevers or endotoxin. 3 major groups of phagocytic cells: neutrophil, mononuclear phagocyte system, organ-specific phagocytes. 3 major groups of phagocytic cells: neutrophils, mononuclear phagocyte system, organ-specific phagocytes. Neutrophils first to arrive, mononuclear phagocyte system, macrophages and monocytes, mononuclear phagocyte system, macrophages and monocyte, organ-specific phagocytes, microglia, organ-specific phagocytes, microglia. How do phagocytic cells get to their target? Bacterial carbohydrates flag the cell for phagocytic attack. Diapedesis get through capillary wall to target. How do phagocytes get to their targets? Bacterial carbohydrates flag the cell for phagocytic attack. Diapedesis get through capillary wall to target. Phagocytes meet particles. Particles become surrounded by pseudopods. And this is with phagocytes. Phagocytes meet the particle. Particle becomes surrounded by pseudopods. Pseudopod surrounds the cell and ingests foreign cells. They're scavenger cells. Pseudopods form a vacuole, what you have once the pseudopod have engulfed the bacteria. Vacuoles fuse with lysomes, which digest the particle. Pseudopods do not fuse, and lysomes can be released into the affected area. Phagocytes meet particles. Particles become surrounded by pseudopods. Pseudopods surround and ingest foreign cells. Forms vacuole, what you have once the pseudopod have engulfed the bacteria. Vacuoles fuse with lysomes, which digest particle. Pseudopods do not fuse with lysomes. Lysomes can be released into infected area. Fever. Cell wall of Gram. Bacteria contains endotoxins. Endotoxins stimulate the release of cytokines. The result of this process is the experience fever, increased sleepiness, and decreased plasma iron. Decreased plasma iron inhibits bacterial activity. Fever, cell wall of Graham. Bacteria contains endotoxins. Endotoxins stimulate the release of cytokines. The result of this process is that we experience fever, increased sleepiness, and decreased plasma iron. Decreased plasma iron inhibits the bacterial activity. Interferon cytokines produce a nonspecific short-acting resistance to viruses. Act as messengers that protect other cells in the vicinity from viral infection. Interferons, cytokines, produce nonspecific short-acting resistance to viruses. Act as messengers that protect other cells in the vicinity from viral infection. 3 types of interferons are A, B, and Y. 3 types of interferons are A, B, and Y. A inhibits replication, increases natural killer cells. A inhibits replication, increases natural killer cells. B inhibits viral replication, increases natural killer cells. B inhibits viral replication, increases natural killer cells. Y activates macrophages. Y activates macrophages, which do what? Only produced by certain lymphocytes and natural killer cells. Y activates macrophages, which do only produce by certain lymphocytes and natural killer cells. Specific immunity, antigens and antibodies. Specific immunity, antigens and antibodies. Antigens are molecules that stimulate the production of antibodies. Antigens are molecules that stimulate the production of antibodies. Antigens combine specifically with antibodies produced. Distinguishes self-antigens from non-self antigens and typically mounts a response only against non-self antigens. Antigens combine specifically with antibodies produced. They distinguish self-antigens from non-self antigens and typically mount a response only against non-self antigens. Major histocompatibility complex. This is a fancy name for the markers I said we find on our own cells. Class 1 are found on all body cells except mature blood cells. MHC class 2 are found on all cells that have taken up a foreign antigen and are presenting that antigen on their surface. MHC, major histocompatibility complex. This is a fancy name for the markers I said we find on our own cells. Class 1 of the MHC complex are found on all body cells except mature red blood cells. Class 2 of MHC complex, class 2 are found on cells that have taken up foreign antigens and are presenting that antigen on their surface. Haptens are small organic molecules that can become antigens if they bind to your proteins. Haptens are small organic molecules that can become antigens if they bind to your proteins. Immunoassay, antigen-antibody complex reactions produce clumping agglutination. Agglutination can be used to assay a variety of antigens. To assay means to look for a reaction, just like we did with the blood typing experiment. Immunoassay, antigen-antibody complex reactions produce clumping agglutination. Agglutination can be used to assay a variety of antigens. To assay means to look for a reaction, just like we did with the blood typing experiment. Lymphocytes are a type of white blood cells. They derive from stem cells in the bone marrow, replace themselves by cell division so the stem cell population is not depleted. Lymphocytes seed the thymus, spleen, and lymph nodes, producing self-replacing populations in these organs. Lymphocytes are a type of white blood cells derived from stem cells in the bone marrow, replace themselves by cell division so the stem cell population is not depleted. Lymphocytes seed the thymus, spleen, and lymph nodes, producing self-replacing populations in these organs. T lymphocytes Lymphocytes that seed the thymus become T lymphocytes. They do not secrete antibodies. They have cell-mediated immunity to destroy foreign cells. T lymphocytes, 65 to 85% of lymphocytes in the blood. T lymphocytes, lymphocytes that seed the thymus become T lymphocytes. They do not secrete antibodies. T lymphocytes destroy foreign cells with cell-mediated immunity. Contact. T lymphocytes are 65 to 85% of lymphocytes in the blood. T lymphocytes and B lymphocytes. What happens? T lymphocytes kill by contact, and B lymphocytes are secreted hormonally. What happens to the thymus as we age? Thymus atrophies, disappears after puberty. Where are T cells produced as an older adult? Nodes in other organs continue to produce T cells on their own. What happens to the thymus as we age? Thymus atrophies, disappears after puberty. Where are T cells produced in an adult? Nodes in other organs continue to produce T cells on their own. Thymus, spleen, and other places. There are three types of thymus lymphocytes, T cells. Killer T cells, helper T cells, suppressor T cells. Three types of T cells are killer T cells, helper T cells, suppressor T cells. T cells attack cells near them. B cells provide humoral immunity, releasing antibodies into the blood where they can travel to the target. B cells provide humoral immunity, releasing antibodies into the blood where they can travel to the target. Killer T cells are released for the attack. Helper T cells regulate the response of T cells and B cells and release B cells. Helper T cells release B cells into the immune system. Suppressor T cells pull everyone off. See, we're good. Killer T cells destroy cells with antigens on their surface, must be actual contact with their victim, defend against viral and fungal infections. Killer cytotoxic T cells destroy cells with antigens on their surface, must be in actual contact with their victim cells, defend against viral fungal infections. T cells secrete perforins that form cylindrical channels through the cell's membrane, causing the cell to die slowly. T cells create perforins that form cylindrical channels through the cell's membrane, causing the cell to die slowly. Helper T cells regulate the response of both killer T cells and B cells. They activate. Who do they have to activate? Helper T cells activate B cells. Why is this not true of killer T cells? How do they get activated? Killer T cells regulate immune system. When CD8 receptor recognizes an infection cell, it activates a cytotoxic T cell. Helper T cells, what do they regulate? The response of both killer T cells and B cells. Who do they have to activate? Helper T cells activate B cells. Why is this not true of killer T cells? How do they get activated? Killer T cells regulate the immune system. When the CD8 receptor recognizes an infection, it activates the cytotoxic T cells. Suppressor T cells indirectly participate in specific immune response, inhibit T cell, B cell activities, affect the amount of antibodies secreted, moderate the immune response. Suppressor T cells indirectly participate in specific immune response, inhibit T cell and B cell activities, affect the amount of antibodies secreted, moderate immune response. Destruction of T lymphocytes. T cells produce fast binds to fast ligand and triggers apoptosis, cell suicide, production of fast increases during infection, destruction of T lymphocytes. T cells produce fast binds to fast ligand and triggers apoptosis, cell suicide. Cytokines that regulate the immune system, interleukin 1, interleukin 2, interleukin 3, and interleukin 4. Cytokines that regulate the immune system are interleukin 1, interleukin 2, interleukin 3, interleukin 4. Interleukin 1, secreted by macrophages and other cells, activates all T cells. Interleukin 1, secreted by macrophages and other cells, activates all T cells. Interleukin 2, released by helper T cells, activates killer T cells. Interleukin 2, released by helper T cells, activates killer T cells. Interleukin 3 stimulates the proliferation of bone marrow stem cells. Interleukin 3 stimulates the proliferation of bone marrow stem cells. Interleukin 4 is secreted by T cells, required for proliferation, clone development of B cells. Interleukin 4 is secreted by T cells, required for proliferation and clone development. What role do these substances play in regulating the immune system? Cytokines. Interleukins help regulate the immune system. I know they play a role in why you get a fever. B lymphocytes, we make them in the bone marrow. Which type of immunity? B lymphocytes are specific immunity. What does this mean? Humoral immunity. What do they combat and how? Provide humoral immunity. B lymphocytes provide humoral immunity. B cells combat bacterial infections as well as some viral infections, secreting antibodies into the blood and lymph that will bind to antigens. B lymphocytes, we make them in the bone marrow. B lymphocytes are specific immunity, and their humoral immunity B lymphocytes provide a humoral immunity, which means that they're secreted into the blood and can go anywhere to fight off the foreign cells that we don't need. B cells combat bacterial infections as well as some viral infections by secreting antibodies into the blood and lymph that will bind to antigens. B lymphocytes, when activated, stimulate a production of memory cells. B lymphocytes, when activated, stimulate a production of memory cells. These are memory cells are considered important for passive immunity. The next time we are exposed to the same antigens, we already have the cells that we need to produce the antibody. We don't have any delay in being able to fight. Memory cells are important the next time we are exposed to the same antigens. We already have the cells we need to produce the antibody. We don't have any delay in being able to fight. And the production of plasma. B lymphocytes are responsible for the production of plasma. Protein factories that produce antibodies, where the antigens will agglutinate. Isolated molecules may be molecules at the surface of invading foreign cells or in the lymph nodes. B lymphocytes and the production of plasma cells, protein factories that produce antibodies. Where are the antigens? And these will agglutinate with. They are isolated in B lymphocytes. Antigens from the protein factories will agglutinate with isolated molecules that may be molecules at the surface of invading foreign cells or in the lymph nodes. Antibody proteins are also known as immunoglobulins. Different antibodies have different structures because they have specific actions. Antibody proteins are also known as immunoglobulins. Different antibodies have different structures because they have specific actions. Antibody structure. What is the FAB region? FAB regions are variable, provide a specific bonding site for antigens. Antibody structure. FAB regions are variable, provide a specific bonding site for antigens. Complement proteins, marked targets activate the complement protein system against specific invaders. Complement proteins, marked targets activate the complement protein system against specific invaders. Complement types, there are 11 complement proteins. C1 is in charge of recognition. C2 through C4 is in charge of activation. C5 through C9 is in charge of the attack complex fixation. Designated C1 to C9. The complement protein types are there are 11 proteins. C1 is in charge of recognition. C1 is in charge of recognition. C2 through C4 is in charge of activation. C2 through C4 is in charge of activation. C5 through C9 is in charge of the attack complement complex fixation. C5 through C9 is in charge of the attack complex fixation. Complement proteins are designated C1 through to C9. Complement proteins are designated C1 through C9. Active immunity. This occurs when we fight things off ourselves. Active immunity. This occurs when we fight things off ourselves. Primary response. With the first exposure to a pathogen, immune response insufficient to combat disease. Latent period of 5 to 10 days before measurable amounts of specific antibodies appear in the blood. Antibodies appear in the blood. Primary response. With the first exposure to a pathogen, immune response insufficient to combat disease. Latent period of 5 to 10 days before measurable amounts of specific antibodies appear in the blood. Secondary response. Subsequent exposure to the same antigen, antibody production is much more rapid. Maximum antibody concentration is reached in less than 2 hours, maintained a longer period of time. Secondary response. Subsequent exposure to the same antigen, antibody production is much more rapid. Maximum antibody concentration is reached in less than 2 hours, maintained for a longer period of time. Passive immunity. When we get immunized, immune protection produced by transfer of antibodies to a recipient from a donor. Donor has been actively immunized, occurs naturally in mother to fetus during pregnancy and mother to infant during nursing. Passive immunity. When we get immunized, immune protection produced by the transfer of antibodies to a recipient from a donor. Donor has been actively immunized, occurs naturally in mother to fetus during pregnancy and mother to infant during nursing. And that's it. Hope y'all enjoyed my last video for my physiology section on YouTube. Peace. Catch y'all in the next one.